Origins of perception. All right, so our journey begins right in the sensory organs. Let's use hearing as our example here. Deep inside the inner ear's fluid-filled cochlea, sound vibrations come in and move the basilar membrane. And sitting right on top of that membrane are these tiny hairs called stereocilia. As the membrane vibrates, these microscopic hairs actually physically bend. It's kind of like wheat blowing in the wind. This mechanical bending triggers the cell to depolarize and boom, neurotransmitters are released. Right there, in that split second, we've just crossed the boundary from the physical world into the biological one.
Now, this crucial crossing point is what we call transduction. It's the fundamental translation of perception. Whether it's mechanical pressure on your skin, light hitting the back of your eye, or sound bending those stereocilia in your ear, the sense organ's entire job is to convert physical reality into the brain's internal electrical language. Moving on to Section 2, transduction and transmission, from ear to cortex.
Okay, so once they're transduced, these signals shoot up into the sensory cortex, expanding across this massive, tangled network of neurons. To really understand how they handle this signal, I want you to think of the dendrites and the cell body, the soma, as the processor. That's where all the inputs are integrated. The axon, on the other hand, is just the cabling. It merely transports this highly compressed, virtual signal, which we call the action potential, to its next destination. The real physical magic? That happens in the processor.
And inside that processor, the generation of an action potential is essentially an anonymous vote. I love this concept. The current arriving at the cell body basically represents a massive tally of all the active synapses. The cell itself doesn't actually know which specific synapse is fired, it just counts up the votes. If enough synapses vote yes, an action potential fires, completely hiding the individual origins of that signal.
Section 3. Generating brain currents inside the dendrite.
Let's zoom in closer on that dendrite processor to see how it builds the sustained electrical signals we eventually detect. When synapses open up on the outer dendrites of a cortical neuron, positively charged ions pour in. This right here creates what's called the primary current, and unlike that virtual signal traveling down an axon, this is a physical, literal river of ions flowing inside the dendrites, heading straight toward the cell body.
But, as we know, biology is wonderfully messy. On its way down to the soma, this river isn't perfectly contained. Some of this electrical current actually leaks out of the cell and spills into the extracellular space surrounding the neuron. And that leakage creates our second major player, the volume current. Because the whole system acts kind of like a battery, it needs to form a complete circuit. So you have the primary current flowing inside the neuron, while the volume current acts as its partner, flowing outside and completing the electric circuit through the highly conductive tissue of the brain. The absolute best way to visualize this volume current is through a nudging analogy. Picture a completely packed room. As positive ions are sucked into the neuron at the top synapses, they leave behind an empty space, right? And neighboring ions immediately rush in to fill it. Meanwhile, down at the bottom near the soma, the ions leaking out are physically pushing against the extracellular ions that are already sitting there. This constant sucking and pushing causes a wave-like nudging effect, literally ions bumping into ions, and that ripple spreads all the way out to the surface of your scalp. Section 4. The neural mass effect. Synchronous activity.
Now, obviously, a single microscopic neuron doing this tiny bit of nudging isn't going to generate nearly enough current for us to measure from outside a thick human skull. No way. But remember the architecture of the brain we talked about. Cortical columns are heavily interconnected. A sensory signal from the ear doesn't just activate one lonely cell, it activates the entire column. We are talking about over 1 million pyramidal cells becoming synchronously active at the exact same millisecond.
And this perfectly illustrates what's known as the neural mass effect. It's not just the sheer number of cells firing, it's their specific architecture. The apical dendrites of these pyramidal neurons are perfectly aligned, running straight and perpendicular to the surface of the cortex. Because they're all pointing in the exact same direction and firing at the exact same time, their tiny individual primary and volume currents actually sum together. So instead of canceling each other out, they stack up to become one massively amplified signal. It's a real game changer. Section 5. Measuring the mind. Decoding perception.
So now we've got these massive summed-up currents echoing through the brain. The big question is, how do we actually capture them and decode human perception without, having to open up the skull? Well, we rely on two incredibly powerful, distinct techniques, EEG and MEG. While both give us millisecond-level timing, they're actually listening to completely different parts of this electrical storm.
Let's start with EEG. EEG effectively measures the voltage differences across your scalp, and it is entirely driven by those volume currents we just discussed. Remember that wave-like nudging of ions? Well, that nudging pushes extra positive ions to one side of the head and pulls them away from another, which creates an uneven electrical charge right on the scalp. By simply placing two electrodes on the head, EEG measures that difference. And because volume currents easily fill up the conductive brain tissue, EEG can pick up activity spreading from virtually anywhere in the entire brain.
On the flip side, look at MEG. Basic physics tells us that an electric current generates a magnetic field, right? Well, MEG completely bypasses those messy volume currents and directly measures the tiny magnetic fields generated by the primary currents inside the dendrites. Because MEG is mostly sensitive to currents running parallel to the skull, and those pyramidal cells sit perpendicular to the cortex surface, MEG is uniquely suited to map activity hiding deep in the folds, the sulci, of the brain. It gives us this beautiful, crisp map of the outer cortex.
It really is a marvel, isn't it? When you think about it, from the literal physical bending of a single microscopic hair cell in your ear to a massive, synchronized electrical storm of 1 million neurons to a faint magnetic field rippling just outside your skull, the journey of perception is nothing short of awe-inspiring. And it leaves us with a pretty compelling final thought. As our non-invasive tools get even sharper and our understanding of this intricate neural architecture deepens, what hidden secrets of the human mind are we going to decode next?